There we go. Okay. So we left off with the discussion, I ended with the corpus luteum. Again, remember that and then we did talk about the corpus albicans and the atretic follicles. For lab purposes, we are not going to be talking about the adrenal medulla, but again, if you would just know that this is the central zone, the inner zone. Again, the term medulla, as you're well aware, is just a generic term that is used to describe the inner zone of any organ or structure. And in the medulla, again, we're going to find a lot of connective tissue. It's fibroelastic connective tissue, very highly vascularized, lots of lymphatic vessels, and obviously it's going to be equipped with some nerve endings. Don't worry about the premenstrual medulla. All right, if you would just simply know that the hilus cells that are present there will secrete some androgens, okay? And it's interesting that if we compare these hilus cells to the Leydig cells located in the interstitial region of the testis, that we see a lot of the same components in the cytoplasm, so similar types of androgen secreting cells. Recall that the Leydig cells are those that are responsible for secreting testosterone in the testis. All right, now the oviducts. The oviducts, I don't care that you know links, and I think that I actually have that highlighted in gray, the 12 centimeters, on your handouts. But again, they're going to have a definitive muscle wall. It's not going to be as extensive as the ductus deferens in the male reproductive system, but the muscularis is going to help you to identify this structure. The wall of the oviduct contains some large veins. Keep in mind that the oviduct is not physically connected to the ovary. It doesn't directly receive the ovum. The ovum is going to be released into the pelvic cavity. And when the smooth muscle of the muscularis of the oviduct, which is also known as the fallopian tube or the uterine tube, contracts, then that is going to constrict those large veins. And the constriction of those veins is going to deform, so to speak, the wall of the oviduct and cause it to be extended. It's going to, that movement is going to move the distal region of the oviduct closer to the ovary. And as we're going to see, the oviduct is lined by different types of cells. One type of cell is a tall cell that's equipped with cilia, and those cilia are going to generate a current and then help to draw the ovum into the oviduct. Okay? All right, as far as the distinct regions of the oviduct, there's 4. The first is known as the infundibulum. The infundibulum is the region of the oviduct that is equipped with those finger-like projections known as fimbriae. And the fimbriae are going to be lined also, obviously, with some ciliated cells. And this is the region that actually overhangs the ovary. Then moving on through the oviduct, the next region is the ampulla. Fertilization of the egg is going to take place in this zone. if you just look at the anatomical diagram of the oviduct in your text, you will also see that this is the widest region, the most dilated portion of the oviduct. Then, as we move on through the oviduct, we'll encounter the third region, which is the isthmus. This is a narrowing region, and then the portion of the oviduct that actually pierces the wall of the uterus is the intramural zone. Now, within each of these regions, we will find the following zones. We're always going to have a mucosa, then we're going to find a muscularis, and then finally a serosa. Okay? Now, there are longitudinal folds of the mucosa in all four regions of the oviduct. Okay? The infundibulum, the ampulla, the isthmus, and then the intramural zone. And we're going to be actually taking a look at the ampulla of the oviduct in our lab when we get to it later on in the lecture today. The folds are more pronounced, much more pronounced in that region, and we're going to see some branching of those longitudinal folds. The epithelium lining the oviduct in all regions is simple columnar epithelium. The simple columnar cells consist of two different types. There are PEG cells. Now, these PEG cells are not going to have any cilia on their free surface. These are cells that secrete a fluid. It's a rather sticky, viscous type of material, and within this secretion, we're going to find some nutrients that are going to help to meet the nutritional requirements of the and metabolic requirements of the ova as well as the sperm as they're moving through the oviduct. Again, keeping in mind that the oviduct is moving the ovum in one direction and the sperm in the other direction. One is coming from the ovary, the other is passing through the vagina, up and through the uterus, and then finally making its way into the oviduct. It's also interesting that the height of the epithelium varies in the oviduct. We're going to find the tallest cells associated with the first segment, the infundibulum, and then we'll see a decrease in height as we approach the isthmus. Okay. Another function of the secretions of the PEG cells that I need to mention is the protective function. That secretion is also going to help to prevent pathogens from actually moving from the uterus up into the oviduct. And now keep in mind that the oviduct opens into the pelvic cavity, so that also the secretion will also help to prevent, again, disease-causing microorganisms from gaining entry into the abdominal pelvic cavity. Second type of columnar cell that we find associated with the oviduct is we're just going to refer to as a ciliated cell. Okay, tall cells with cilia. These are the cells that are going to generate currents and move the sperm. They're responsible for the movement of the ovum, and they're also going to aid in the movement of the PEG cell secretions. Okay. Lamina propria. All I want to mention about the lamina propria of the oviduct is not that remarkable. It's just a regular connective tissue or loose connective tissue, as we've seen in other in other organs, just a loosely organized connective tissue. All right, now the muscularis, and I think that the green text I have either omitted in your handout or have it grayed in. Is that correct? Because that's material that you're not responsible for, anything in green. The muscularis, there are two layers. We're just going to identify the muscularis as such for lab. I would like for you to know that there are two layers for lecture purposes. There's an inner circularly oriented layer of smooth muscle and then an outer longitudinally oriented. Now, when I say that it's poorly defined, I mean it's very difficult to, no, that was not extreme. it's sort of difficult to distinguish between the two layers, okay? You can see it. All right, now the external surface of the oviduct is going to be covered by a serosa. The composition of the serosa is essentially like that we've encountered covering the outer surface of other organs. We're going to find a layer of mesothelium and then a thin underlying layer of connective tissue. Okay, this is a nice section through the wall of an oviduct. This is the luminal surface. This is the external wall. Notice that we find these projections of the mucosa, these folds, and notice that the folds branch. Okay, this is actually a section taken through the ampulla, and notice again the significant branching in this zone. The muscularis, this 2 layers of smooth muscle is here, but again, in some regions it's difficult to discern where, the difference between the orientation of the 2 layers. And then we see the serosa. This is obviously a low magnification, but if you can see right there, there's the nucleus of a mesothelial cell. There's one, there's one. So we have a layer of mesothelium with an underlying layer of connective tissue. Okay. Another section through the oviduct. This is much better. Again, the luminal surface, the external wall. Notice the projections of the mucosa. They branch here. The muscularis, smooth muscle here. Okay. Now, this is a high magnification of the mucosa, through the wall, so we can see the mucosa, the lamina propria, and then the simple columnar epithelium lining the surface. And these cells that stain a bit more intensely are the PEG cells. And notice that the PEG cells lack cilia. And then we see the ciliated cells. I think I have a higher view. Yes. Here we find our PEG cells, our tall columnar epithelial cells. Again, the cytoplasm is going to stain a bit more intensely. It's more basophilic than that of the ciliated cells, and they lack this cilia on their apical surface, whereas the ciliated cells that you see here, which are also columnar cells, are equipped with cilia on the free surface. And we see the lamina propria lying beneath the epithelium. Okay, more sections taken through the folded mucosa of the oviduct. Again, notice our PEG cells, the ciliated cells. Okay. So the oviduct, the intramural region of the oviduct, pierces the wall of the uterus, and the uterus is commonly referred to as the womb. It's roughly a pear-shaped type of organ with an extensive smooth muscle wall known as the myometrium that we'll consider in a moment. The 3 anatomical regions of the uterus are the body, the fundus, and then the cervix. The body of the uterus is the location where the intramural portions of the oviduct penetrate. The fundus is a rounded base of the uterus, and it actually projects above the openings to the oviduct. And then the cervix is the portion of, it's a narrow portion of the uterus that actually is going to project into the vagina. In our slides today, the lab slides, we'll take a look at a section through the cervix and vagina, and you're going to note an abrupt epithelial change in the cervix as it protrudes into the vagina. Okay, so let's go through the wall of the uterus. There are 3 layers we'll be considering. The lining is known as the endometrium. Then there is a smooth muscle layer, the myometrium. Well, I guess we need to talk about the endometrium first, don't we? The cells lining the uterus are going to be simple columnar cells. The exception to this is rule it will be when we're taking a look at the cervix, where the cervix actually penetrates and protrudes into the vagina. We're going to see a change in epithelium to stratified squamous. Now, the epithelial cells lining the endometrium are not going to contain any basal cells. Are all going to be just these tall, simple columnar epithelial cells. And as in the oviduct, we're going to find that certain cells are going to be equipped with cilia, and we will find some non-ciliated secretory cells as well. Again, lamina propria is composed of loose connective tissue, and in the lamina propria we're going to find the uterine glands. The uterine glands produce a product that is going to contain, again, a source of nourishment for the fertilized egg or blastocyst, which is an early developmental stage in humans, until the tissue fully embeds within the wall of the uterus. If we break down the zones of the endometrium, we'll find that there are 2 distinct regions. The outermost region that's actually borders the lumen is known as the stratum functionalis. This is the layer that is going to be actually detaches and sloughs away, passes out of the uterus through the vagina during a woman's monthly menstrual cycle. There's a constriction of the blood vessels that serve the stratum functionalis that occurs during the uterine cycle, and eventually the lining cannot be maintained and it detaches and sloughs away. Lying beneath the stratum functionalis, we find the stratum basalis. The stratum basalis is going to contain many more fibers than that we find in the stratum functionalis. And for this reason, the stratum basalis is going to stain more intensely. The stratum basalis is going to proliferate during the proliferative phase of the uterine cycle and act to repair or rebuild the stratum functionalis. Okay. The myometrium. Again, the myometrium is a thick, smooth muscle layer. It's interesting that the size of the smooth muscle fibers will vary depending on the levels of estrogen. The more estrogen, the larger the cells. Less estrogen, the cells are smaller. And when estrogen is the female hormone that's required not only for the development of the female reproductive organs, but it's required for the maintenance as well. So when if the estrogen supply is extremely low or essentially nil, then those smooth muscle fibers will atrophy and will actually observe some cell death in that region as well. As far as the number of layers of smooth muscle cells associated with the wall of the uterus or the myometrium, there are 3. There's an inner longitudinally oriented layer, a middle circularly oriented layer, and then an outer longitudinally oriented layer. Very extensive layer. All right. The outer layer, the middle layer of the myometrium is extremely vascularized. Okay, and we're not actually going to be identifying this zone for lab purposes, but this region where we see a high degree of vascularity is known as the stratum vasculare. Okay. Now, the lining of the cervix is, the endometrium of the cervix is not sloughed during the menstrual cycle. And if we look at the myometrium, the smooth muscle layer is not nearly as extensive, and eventually it's going to just be replaced by some fibroelastic connective tissue. Okay? All right. Looking specifically in the cervix, as I just mentioned, the myometrium eventually is going to be replaced by this fibroelastic connective tissue. And let's see, don't worry about, see, increase in uterine size. Mark that out of your notes. Did I even include it in your notes? Good. If it's not in your notes, you're not responsible for it. I don't care that you know about the smooth muscle cell population, blah, 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 and I don't care that you know about unclear whether division due to blah, blah, blah. Okay? Don't have time to do it all. So, just the first bit of information that you see there. We're not going to worry about this. Okay? We'll just look at the histology in a non-pregnant female. Okay? All right. Now, the outer layer of the uterus will vary, again, depending on whether it's bordering the pelvic cavity or if it is actually associated with the body wall. And the uterus itself is going to be slightly tilted. It's going to tilt anteriorly, and when it does this, it's going to actually come into contact with the bladder. So in that zone, we're going to find that the wall is actually, outer wall is going to be composed of an adventitia. But all other regions of the bladder will be covered by the serosa. Okay. This is a low magnification of the uterus. Again, we're going to, you would be expected to recognize it at a higher level of magnification. But I just wanted you to, come on, there we go, just to show you how extensive the muscle layer is. The endometrium simply consists of this zone right here. This would be the luminal surface, and then the myometrium consists of this layer here. Very extensive. Now we're looking at the endometrium at a high power. Okay, let me just get you oriented. I'm tracing the junction between the endometrium and the myometrium. This, to the left, we find the endometrium. To the right here, we find the myometrium, the smooth muscle. Now notice how much more intensely this zone of the endometrium is staining. This zone represents the highly fibrous stratum basale, okay, and here we would find the stratum functionalis. Okay, stratum basale, stratum functionalis. Again, this is a section taken through the uterus. I'm going to ask you to distinguish between the phases of the uterus, the proliferative, secretory, and menstrual phases in lab only. So I'm just pointing out that this is a section taken from a uterus in the secretory stage, and all of these little structures here represent the uterine glands. And notice how they've enlarged a bit, hypertrophied a bit. They're being stimulated to up their secretions by the female sex hormone progesterone. So the glands enlarge. Okay? Again, let's just, for our purposes for lecture, this is a section taken through the wall of the uterus. Here we find the luminal surface. The smooth muscle layer or myometrium would begin here and extend down. The endometrium is this zone here, and then once again, notice this intensely staining stratum basalis, lots of collagen fibers, thicker bundles of collagen fibers in this zone compared to the stratum basalis here, lighter staining. And all of these structures represent the uterine glands. This is a slide taken from a uterus in the proliferative stage. The proliferative stage follows the menstrual stage. This is the time period during which cells in the stratum basalis are dividing and replacing the cells of the stratum functionalis. Okay, again, more of the same. This is, we're going again, for lecture purposes, I simply want you to describe the wall of the uterus, say during the proliferative stage, okay? But we're not going to worry about the secretory stage and the menstrual stage for lecture, okay? But again, uterus, luminal surface, myometrium, stratum basale, stratum functionalis, and the uterine glands. Okay. And this is a section taken through the entire wall. The serosa would be here. Again, we are not looking at it at a high enough magnification to actually view the mesothelium, but extensive myometrium, endometrium, basalis, functionalis. This is the secretory uterus. Again, myometrium, endometrium, basalis, functionalis, and look at the size of those endocrine glands. All right, cervix. Just want you to note that the epithelium is going to change from simple columnar epithelium to stratified squamous as it enters the vagina. And the remaining wall, remember how we talked about how the smooth muscle is going to be replaced by a dense fibroelastic type of connective tissue. And as I pointed out earlier, the mucosa of the cervix is not going to undergo changes in the menstrual cycle that lead to detachment. Okay, the lining of the cervix remains intact. Okay, it doesn't slough away. And then the last structure associated with the female reproductive tract that we'll consider is the vagina. The vagina serves as the birth canal and also as the receptacle for the penis. I don't care that you know the length of the vagina. If we're looking at the proximal region of the vagina, we're going to find three different layers. Once again, a mucosa, a muscle layer that we always just refer to as a muscularis, and then an adventitia. The epithelial lining of the vagina is stratified squamous epithelium, non-keratinized. We're going to find Langerhans cells associated with the epithelium in this zone. Again, this is a port of entry into the body, and the Langerhans cells are defensive cells, antigen-presenting cells that help to protect us from disease-causing microorganisms. This is a section taken through the wall of the vagina, and here we have the mucosa, the muscularis, and adventitia, and at a higher magnification we would be able to view the stratified squamous epithelium. Again, this is the lining of the vagina, the stratified squamous epithelium, non-keratinized. Notice the nuclei in the surface cells. In our lab, we'll be looking at a slide that's taken through the cervix and the vagina. It's a longitudinal section. So you will be able to identify the cervix and the vagina and distinguish between the two, both by the projection of the cervix as well as the change in epithelium. Lamina propria, again, not that remarkable, loose connective tissue. We will not find any glands in the mucosa of the vagina. Muscularis, again, there's two layers of muscle. There's an inner circularly oriented smooth muscle layer, an outer longitudinally oriented layer, and then a ring of skeletal muscle that functions as a sphincter at the external opening of the vagina. Okay, but two layers of smooth muscle. And then the adventitia, again, just dense fibroelastic connective tissue as we've encountered with other organs. Okay? Questions? All right. Now, I recorded this lecture also. I'm going to start a separate recording for the lab so that it will not be as lengthy. All right.